It's my great pleasure and honor to introduce Peter de Keizer, who is an academic and also an entrepreneur. So he is an associate professor of senescence in cancer and aging at UMC Utrecht. And at the same time, he is the founder and managing director of Cleara Biotech that he founded in 2018. It was covered in Nature Biotech recently in a really nice article by Ali Dolgin titled Send in the Senolytics. And his work is focused on senescence and peptides that can hopefully kill the senescent cells. So I would not take much more of your time, so the floor is yours.
Thanks, Alex, for the kind introduction. So indeed, I didn't take the easy route. I tried to combine two careers at one. And that's because I think it's good if biologists are able to dig into the mechanism of a process such as senescence, then to try to develop their own compounds and try to get it to the clinic. So the AR part of my talk today is talking about senescence, but then not so much about senescence as a whole, because when we talk about things like cancer, we also don't talk about cancer as a whole. We talk about subtypes, ALL, melanoma, glioblastoma, etc. And I will argue that for senescence, I think we should move in that direction too. For instance, you see here on this slide all senescent stainings, and you can see that they don't overlap within the same cells. I'll explain this in a minute in more detail. The DD part is about the company, so how we are able to design compounds against individual subtypes of senescent cells, and one of these are phospholipeptides we're trying to translate into the clinic, and I'll show you some data on that. As you may recall from the talks from yesterday, from Jim Kirkland, and also earlier today from Manuel Serrano, for instance, there are many hallmarks of senescence. And if you recall the work from yesterday, that slide from Jim, it had so many arrows. There are many hallmarks of senescence, and that also complicates the whole thing. There's no real agreement on what senescence is. So what we do tend to agree on is if you just have healthy cells, here depicted on the left, and one becomes senescent, it gets its S-beta-gal positivity, it starts secreting things, and it affects its neighbor. That's as far as I would like to go on something that is true in almost all scenarios. The fact that SASP as a whole is deleterious was also nicely shown by several papers. Again, if we look at the work from Jim Kirkland, who presented yesterday, they took young mice, injected senescent cells in the fat pads, and you have aging in other tissues. So there's communication from senescent cells in one tissue to another tissue, in this case muscle and brain, and you get frailty. Other work, for instance, done by a colleague of ours in Groningen, Marco De Maria, at that time at the Buck Institute, he nicely showed this for cancer. If you implant cancer cells in a mouse, here indicated with the bioluminescence imaging, you see that they will actually progress to liver and lungs if you give chemotherapy. Chemotherapy, we now know, induces cellular senescence in these organs. So ironically, whereas the chemotherapy kills the primary tumor cells, it promotes metastases. But if you now activate a kill switch, which was specifically present only in the mice, so not in the tumor cells, you see that you prevent metastases from occurring. In other words, senescent cells do something to the environment that can attract cancer cells and make them grow happily. And this is something I'll talk more about in light of the company that we started. So there is something as the SASP, but what is the SASP? Is it IL-6? I have to admit I'm guilty also for looking mainly at IL-6. Also the previous speaker talked about it in the context of reprogramming a lot. There's of course many more processes that regulate IL-6, but it's at least one of the factors that is most predominantly elevated by senescent cells. In 2017, we showed that there's a possibility for killing at least some types of senescent cells in mice by interfering with the process of FOXO4 TP53. And both proteins are very important, so you cannot impair the signaling of both proteins completely. But if you break this one interaction with a peptide, we can actually partially rejuvenate these fast-aging mice, shown here on the left, the TTD mouse model. And if you look at individual organs, we observed, for instance, a drop in interleukin-6. So again, the TTD mice show in green a lot of IL-6 in their kidneys in this case, and other organs as well, and the FOXO4 peptide could reduce that. So is then IL-6 equal to the SASP? That's then the big question. And you can argue, why care? You know, the SASP is a broad definition. It has many components. Does it really matter? Well, It does, because if you look at this slide, this is something that concerns me in the senescence field, why we should do a better job at defining different types of senescence. If you look at the viability of senescent cells, in vitro in this case, in red, to our FOXO4 compound, versus control cells in black, you see that there's about 70% elimination of these cells. If we co-stimulate these senescent cells with IL-1 alpha or beta, which are two SASP factors, the response is much better. Whereas if we use the receptor antagonist, the response is blunted. So these are still senescent cells. So technically, I didn't do much. According to the old school definition of senescence, they're still senescent cells, but the state in which they reside matters a lot. So how does this work in a human being? If we start looking for senescent cells, there's no such thing as senescent cells. Important. So what defines senescence? Again, we had a whole talk about it yesterday with a lot of arrows, and I just summarized a few of the most popular parameters that we like to use. Loss of proliferation, for instance, EDU and Ki67, acid beta-gal positivity, nuclear P16, P21, so they're usually mutually exclusive. I like to work a lot with lemon B1 loss, very potent marker of senescence. And then you've got the famous TAFs, the telomere-associated foci, which usually overlap with what is called DNA scars. And then there are these SASP factors that are being secreted. And there are many. There are over 50. And Jan van Deursen at some point mentioned there are 500 if you take along not just proteins, but also microRNAs, lipids, and metabolites. So SASP factors that are popular to look for are IL-1, IL-6, and in our lab also MMP1 and MMP3. But then, what defines senescence? Is it any of these, all of these, a mix of these? And then they don't overlap, so what is then the definition of a senescent cell? We all do something different. Some people do acid beta-gal and they say senescent, some people do P16, some people just look at IL-6. So I think we should do a better job and define subtypes of senescence. For me, what kicked this off was an observation I made myself as a postdoc, so that's been a while. I don't do too many experiments myself anymore. But here I did two stainings of famous senescence markers, acid beta-gal, which is actually a blue staining, and I just made a gray image that I merged with the other staining, IL-6, in red. You can nicely see that if you would compare this to control cells, those would be all at 0, 0, so they are negative for both. So these cells are all positive a little bit for both markers, but if you do an overlap, you can nicely see that these are mutually exclusive. If we would do a qPCR, for instance, for a lot of senescence markers, we would argue they're all up. IL-6 is very much up in its population, but clearly there's only a subset of cells that produce the IL-6. This is not a technical artifact, because that you can argue acid beta-gal is an enzymatic reaction, IL-6 is a staining. We also did acid beta-gal with P21, and you nicely see, for instance, cells which are, I don't know if the mouse pointer works, but yeah, I think it does. So you see, say, for instance, cells that are blue, no, you should not press it. So that are blue but not green, that are green but not blue, that have both. So there is heterogeneity for both markers. You see the same with the SASP factors, IL-1, IL-6. They are not in the same cells. You have, if you look at this population, only actually very few cells produce IL-6, whereas they're zapped with 15 grays ionizing radiation. They're 100% senescent cells. Also MMP3, also a famous one we work with. Again, that usually clusters together. You see groups of cells producing MMP3. We should not ignore this. And this is important, because if we want to know for a particular age-related disease in mice or in humans, what senescent cells are the causative factor, we need to know which subtype we should be looking for. And then, of course, like I showed you in the previous slide, targeting by phosphopeptides is heavily affected by which SASP factor we co-stimulate with. So for all these golden bullets out there, like Q and D or fisetin that we heard yesterday, I don't believe that, to be honest, that they will work against all types of senescent cells under all conditions. We need to do a better job. If you look at how people do it in the literature, if you go back to the original publications in 2008, when the SASP was first coined, you see, for instance, by this protein antibody array from Judy Campese's group, in the bottom you see healthy control cells, they're all blue for these factors, and in the top you see the senescent cells, fibroblasts in this case. They're all positive for these factors, and there's about 25 in this slide, but now we know there are many more. This is antibody arrays, and it seems then that everything is always up in senescence. It's not true. If you look on an mRNA level, you see that there is a timing event. So indeed, the longer you wait, the more you get a shift towards certain types of cellular senescence. But as I showed you in the previous slide, if you go on single cell level, you find that IL-1 and IL-6, which are here both elevated at 144 hours are only expressed in 20, 30, 40% of cells, so not at 100% at all. In other words, when I see these things in literature on mRNA or antibody level, I always worry like we're missing things. So this is bulk analysis, and we should look at single cells. And that's done challenging a paradigm, which I hope to actually publish on soon. There's no such thing as senescence, or at least no such thing as just senescence. We should define subtypes, like we do in cancer. So there's things subtypes. And then just an example of how you can do this, if you look at data sets of single-cell imaging, this endothelial cells that were negative for LMB1, so also I here am guilty of using a marker that I like to work with, LMB1 negativity, in this case also negative for HMG1 and positive for P16. Even there you can already identify 4 subtypes by single-cell RNA sequencing. We also invested heavily in another technology called imaging mass cytometry, where you now can actually combine up to 4D markers at once, so we not rely on immunofluorescence, and you can do this on tissues very well. So right now here we show you 9, and they're not all senescence markers, but you can nicely see, for instance, that the smooth muscle actin positive cells are also PML positive, and then MMP2 and 3 actually co-localize with another subtype. So we are now using this, we are expanding the panel. We have now 20 markers up and running at once. Again, we try to make this 40 with cell-based markers, and then we want to test this in aging. The downside is this is disruptive, so you destroy the tissue, so you cannot reuse it anymore. The challenge is that the correlations can become enormous. So Alex, if you're listening, artificial intelligence is welcome here. So that's that on the aging part, on the AR part. Then the DD part, put your money where your mouth is, I always say. If you think that there are these subtypes and you think you can target them, Jim said yesterday there are over 100 senolytics, so why bother? And why has it not been progressed into the clinic? Well, I moved into drug discovery. That's a totally different world, I can tell you, and there are many more quality control issues that you have to abide by. So our FOXO-PF3 inhibitors are new drugs. They're not generic, they're not repurposed. That means we have to do all sorts of new pharmacology and tox studies. So this is part of the company that we founded to actually progress these compounds towards clinical trials. This is a slide just in vitro to show you how we improved one compound. We tested around 400 new compounds by now. The old version that we published in Cell, we call it 3001, so clearer drug, third generation first compound. You see in the top the selectivity versus a subtype of senescent cells in irradiated RP1 cells in this case. And if we compare it to the new compound in the bottom graph, you see that the compound 183, so clearer drug, fourth generation, number 183 has a very strong selectivity against these cells. CL04-183 eliminates these senescent cells not through necrosis, not through necroptosis, ferroptosis, etc. It is clean apoptosis because it shows caspase activation in these cells, and we can block it with caspase inhibitors. Another thing we worked on a lot, which is not necessarily maybe when working with generic compounds, is stability. So peptides have a reputation of being unstable, and you can see, for instance, in red, the original peptides from the Cell paper, stability in hours. Half-life was about 50 hours or so, but now we optimized it in such a way, because we identified the breakdown products, to make it stable for over a month. So 2 months on the bench and still very little breakdown. Then, the thing which will bore all of you as academics, but from a pharma point of view is very interesting, and this is what shocks me a little bit when people are doing clinical trials with generic compounds, and also what Jim was saying yesterday with the Q&D, 20 clinical trials, whereas they don't know the mode of action of these compounds. They have never done maximally tolerated dose studies as far as I know, and we don't know the PK. We don't know in which tissues the compounds end up with. Because it's a new drug that we are working on, have to do all these things. So we did maximally tolerated dose studies, we did pharmacokinetics and pharmacodynamics, and we showed that there's a favorable uptake from plasma to tissues of drug 4183. There's no toxicity in vitro in cytokine release. We did mass spec-based pull-downs and proteomic analysis, show no major tox pathways activated. We have extended stability in plasma, liver enzymes in solution, and in a very strong cellular uptake. Then we decided, well, what do we want to use these compounds for? Do we want to go for aging, age-related diseases? It's all very difficult. You know, Unity's clinical trial was based around questionnaires, and they failed because these are quite soft endpoints, and a lot of things in aging are quite soft. So we chose to go for something very hard, and that's, in this case, oncology. And you can say that's not what we're here for. I mean, this is an aging community. But I think for getting something into the clinic as a new compound, cancer is actually great because you have very hard endpoints and you can actually do very good biomarker research. So in Utrecht, we are all very heavily involved in 3D cultures, 3D colon cultures especially, but also liver and kidney, for instance. And you can get a lot of these straight from the clinic. So if we take 3D cancer patient organoids, and we give, for instance, very high doses of the standard of care, 5FU, we see that these are insensitive to these drugs. So those drugs are cytostatic, but not cytotoxic. And you can see in this live-dead assay that the cells remain green. If you now apply the compound of ours at a very low dose, actually, you see that this completely wipes out the cancer patient's 3D organoids, and no effect is seen on wild-type organoids from the same patient. We also measure this in terms of caspase activation, and you can see that caspase activation occurs within a day, so it's a very rapid response, which is also very important if you want to go for targeting, because that means you can do bolus injections in patients eventually. And if we compare it to the old compound from the Cell paper, we see that the 301 does not effectively eliminate these colon cancer organoids, whereas the new compound does. We don't see that there's some mouse experiments. Now we have many more results in larger cohorts of mice, but because this is company data, that's proprietary. It has not been released to the public yet. So this is a figure I can show where we have, again, we have a PDX with metastatic colon cancer implanted in the cecum of the mice. They metastasize throughout the animal, and this is very nicely inhibited by the compound. And this is because we're actually inducing apoptosis in these cells, so they are positive for the markers of senescence in the cancer cells. So these are cancer cells positive for the biomarkers of this subtype of senescence. And you can actually see that only in the human cells we induce tunnel positivity, so you get apoptosis. So it's not because we stall the cells, we actually kill them. And the last thing is then we also did this in other types of cancers, several in fact, but let's say here metastatic breast carcinoma. We grow these tumors for up to 5 weeks on the flank of the mice, then they start to metastasize. Not all of these mice do, so this is a model where it's about 50% of the mice that develop spontaneous metastases. But this is all inhibited with the compound. Again, we now have in very large, statistically significant cohorts. And then the interesting part also for the senescence field is that if you look at fibrosis and stromal infiltration, you see that in the lung and the liver, these tumors actually infiltrate the stroma. So you get actually, it's quite interesting that the tumor cells make the stroma more senescence-like. And this is something which we can undo with the compound. So in both the lung and the liver, you see that there's a nice opening up of the lung if you give the 4183 compound. And also in the liver, you see that there's now a very hard divide. I know it's very visible in the bottom right, hard divide between tumor and stroma. And if you do biomarker staining, you see that we lose the biomarkers of senescence in these tumors and in the stroma. So the conclusions for this ARDD meeting are as follows. For the aging research part, I showed you that senescence is not a definition at all. It is actually a mix of subtypes. So you have different types of senescent cells, and probably I think we should work on compounds to target these individual subtypes. And I don't mean that a senescent hepatocyte is different than a senescent fibroblast or a senescent melanocyte. No, I mean within the same cell types you can even have heterogeneity. So intra-cell type heterogeneous effects and drugs. Many SASP factors are only expressed in a subset of senescent cells, only 20–30%. qPCR would show you that they're all elevated, but not if you do immunofluorescence or IMC. So IMC and single-cell RNA-seq are now under development in our lab to do this in tissues, and there also we showed that in human samples there's a lot of heterogeneity that we can detect. And again, I'm a bit fascinated by the whole idea of senescent cells going to humans, but I think we should do a better job in defining then which compound is going to target which subtype. So that brings me to the DD part. We chose to optimize our FOXO-P3 compounds for one of these subtypes. 4183 is a 4th generation. We now also have a 5th generation peptide that we will likely progress into the clinic. We have PK, PD, ADME, safety, toxicity, maximally tolerated dose, all these things which you as an aging community don't care for, but which are important in drug development were done, which were never done for QND or navitoclax as far as I know. And I think therefore it took us a lot of time, but we're doing it at least the right way. And then we are able to use them now against colon cancer and memory carcinoma. And we have chronic disease programs ongoing, and I'm happy to tell you about that in the coffee break. And thank you very much for your attention.
Are you there, Alex?
Yes, I'm here. Can you hear me?
Yes.
Perfect. So thank you, Peter. Very happy with your lecture. It's truly amazing work. And we are all routing for Senolytics, hoping to see them in the clinic as soon as possible. So we have very few minutes left for questions. Let's do one and then transition to Slack, which is populated with a lot of questions. So a question from Morton. So IR-induced senescent cells are probably different from oncogene-induced senescent cells. Which types are most easy to target with FOXO4 DRI?
Yeah, so this is not entirely understood yet. So we think actually it's what we call scarred cells, and they have a lot of DNA scars, obviously. And oncogene-induced senescent cells are a sledgehammer, so they have everything on. So there, if you would do a staining for the SASP factors, they're all very heavily elevated. Of course, there's no such thing as only OIS, because I'm talking now about RAS-induced senescence. If you have BRAF-induced senescence, you don't have the DNA damage response either. So again, different types of senescence, different types of responses, and you need to know which type of response your compound is best at. So, yeah.
Thank you very much for such a detailed answer. And there are more kind of larger and more philosophical questions out there in the Slack. Would highly appreciate if you can answer. And just very brief question. So how far are you away from, let's say, IND submission?
Yeah, very good question. That's one we get all the time. We hope to be within a year and a half from IND. We are hope also to announce not too far from now that we have a major step up in this. Not yet. It's not yet fully concluded, but we're quite advanced in raising more capital to progress the preclinical and clinical development plans. Yeah.
Perfect. Well, there are lots of venture capitalists in the room, so you might be able to find some interesting people. And
we have one question here in the room, Alex, also, that we'll take real quick.
Yeah, great talk. I had one question for your chronic disease program in aging. Yeah. Would it be important to also test toxicity in aged mice with this new compound?
Yes, of course. So we have done studies in aging, but indeed, MTD studies usually happen in very young mice and usually SCID mice. So yeah, arguably you would have to do it in Black 6 or FEB and then also in aged mice. But doing that at a CRO is extremely expensive. So this you only do when you really have an indication that your compound is it. So this was one compound that I showed you. We're progressing that for cancer. For that we don't need a new MTD, but we have another compound for the chronic disease program, and there indeed I think you have a valid point. And I would hope that QND would, and navitoclax would also at some point be tested in those. But, you know, expensive.
So thank you, Peter. I would love to see you on Slack.